Well, thank you very much for the nice introduction, and it's nice to be back at Tel Aviv. I was here for a while in 1982 with Yigal Koltin, David Gutnick in a department that I understand doesn't exist anymore. Like many things, sic transit gloria, right? Microbiology is gone. And I want to thank also especially the David family and all the people who gave us this really nice award. So I was asked to give a talk on science, and I took it very seriously, and that's what it's going to be. And it's not about me. It's really about a question that one of the students asked me this afternoon about what is the proper role of model systems relative to direct study of humans. And actually, I told her, and I meant it, that my talk this afternoon is in no small part about that. Okay, so here's the argument. I can't see the slide, all right. So you can read the English. Basically, the real take-home from genomics, the top-level result, is about evolution. That the sequences show us that the similarity in the individual proteins and the protein functions can be traced all the way back to the common ancestors of all the biota on the Earth. And what that says is that we can actually learn in one organism where it's convenient what a protein does and be not certain but highly confident that it does something similar if we have the same protein. The cells that, epithelial cells, let's say, that have gained the activity of a particular, of the ability to grow when they shouldn't, can evolve into, is there a problem?
No, carry on, I'm sorry.
Okay. I'm easily distracted, I'm afraid. Okay. So the analogy that I'm trying to give you is that if I grow yeast and they grow better, then this is a very similar problem to that faced by a epithelial cell that has just learned to grow in where it shouldn't, and as a consequence of its ability to do this, it can evolve. And the question is, since there's conservation of the proteins themselves, can there also be conservation of the mechanisms by which evolution occurs? Okay, so I'm going to start with this wonderful picture from our friends in Toronto, and basically this is the universe of function. Each of these dots is a protein, expressed gene, and the distances between them are calculated by a very simple algorithm that takes the phenotype of double mutants and asks, is it different from what you expect? And if it is, then the two genes are said to be related in some way, and if it's not different, they're not related. And then all of this stuff automatically generates this figure. And what's amazing about this automatically generated figure is that the colored areas, if you look in the gene ontologies, make sense. They actually fall by themselves just by this very simple idea. They fall into things having to do with mitochondria or peroxisomes or DNA replication or vesicle-mediated transport. In other words, we actually have some idea of the system relationships just from a very simple thing. Now, the thing, of course, is that the very simple thing works in yeast, and many other very simple things that were tried in yeast don't work nearly as well as this, but it is an existence demonstration that there are relationships and that these can be discovered by genetic tests. So these gentlemen were, how many of you have heard of Jacques Monod before? Fraction. Okay, he was the parent of microbial physiology, and the famous lactose operon and inducibility of genes. And how many of you have heard of Leo Szilard? Many fewer, no surprise. Leo Szilard was a minor 20th century physicist whose main claim to fame was that he invented the atomic chain reaction and the atomic bomb and got patents on both. After the war, he decided he didn't want to do atomic bombs anymore, and he invented, along with Monod, this gadget called the chemostat. And the chemostat is a device in which bacteria or yeast or any free-living microorganism can be grown continuously in steady state. And the idea is simple. You have a huge vat of fresh medium. The medium drips in slowly. We use pumps, of course, and lots of instrumentation I'm not showing you. And then the finished culture overflows. And it's very easy to persuade yourself that in such a system, we can decide what is the rate-limiting nutrient. We just take out, let's say, phosphorus until they can't grow to as high density as they used to grow. And secondly, these things are a primary selection for better growth on the limiting nutrient. The obvious way for an organism to grow better on limiting phosphate is to figure out better how to grow on phosphate and not the rest of its functions, because phosphate is limiting. When we did experiments in the early, about 2000, and we did this with glucose limiting, we found a result which, like the Costanzo et al. Result, is remarkable because it's interpretable. What we did was we had 3 different runs of this, and in each case what we discovered was that this was glucose limiting, was that whereas the normal yeast gets most of its energy by fermentation, okay, which is to make alcohol, which is, after all, why we love yeast, right? And the evolved strains, when we fished one out, they turned out to have turned down the flux through this pathway and instead used the oxidative pathway. In other words, they read the textbook. Those of you who've taken biochemistry will know that the biochemistry textbooks, for better or for worse, say that you get 2 ATP per molecule of glucose going this way, and you get 30 plus. Actually, every edition of Streyer's book has a different number for this, okay? But 30 plus ATP for going this way. Okay, this obviously raises the question of why do they do this inefficient thing? Maybe it's not so inefficient if you think about it, or maybe they're just in chemical warfare with their neighbors. Not clear. I'm not here to tell you about that, but I'm here to tell you that I do this evolution experiment, thousands of generations, and I get this result. In other words, it's also a statement about the system. It says that when I'm limiting in glucose, I have a limited palette of ways in which I can get better. Okay? Of all the 5,700 genes, there are only a few that I change. And here is an idea of what the data look like for this. As you can see, here are the respiration genes. They're going up. Red is up. The fermentation genes are going down. And like very good politicians, the transporters are divided. Okay. When we did this experiment again in diploids, we discovered that we sometimes got genome rearrangements. These are plots of the relative amounts of each gene relative to the genome, and the gray parts are 1, and this is 2 or more, and this is less than 1, which is usually 0. Okay? And you see that there are copy number variation, and I know there's a big interest here in Israel as well. And what we discovered was that the copy number variation wasn't random, but in fact, frequently we've got the same rearrangement in exactly the same place. In this particular place on this chromosome, you can see there's extra copies here, and there's a deficit over here, and the break point is always in the same place on the genome, and it's right around this transposon remnant, and here's the data in detail, and the take-home lesson is that this thing is CYT1, and CYT1 is the first step in the oxidative pathway. So why is that important? Well, what we think is that the promoter for CYT1 has been exchanged with some other gene, which is probably expressed better. You get more CYT1, you get more carbon going into the citric acid cycle. Why am I telling you this? Oh, well, we did it, of course, that's one reason. But the other is because of the analogy with the Philadelphia chromosome. Philadelphia chromosome was the first chromosomal rearrangement in cancer that was documented, and what happens in the Philadelphia chromosome is that this gene on chromosome 9 is exchanged, has its which is a growth-promoting gene, which is normally turned off in the cells that become cancerous, gets switched with a gene that's expressed in that cell type, and the rearranged thing, BCR-ABL, provides growth promotion when it shouldn't and is the cause of acute myelogenous leukemia. This is called the Philadelphia chromosome for historical reasons, and for that reason, I like to call this the Palo Alto chromosome. Okay. You will see, however, that the city analogy is all very nice, but there's got to be some underlying understanding that in yeast and in human cancer, the same evolutionary process is at work. We've done many more of these. Many of these make sense. And we've ended up with 24 of these relatively heroic experiments done mainly by Maitreya Dunham. And what we've done here is we've looked at genome-wide gene expression of different selections that we did on glucose limiting or phosphate limiting or sulfate limiting. And we got a very nice result here, which is that for all the sulfate limiting ones, they're all more similar to each other than they are to the rest of the diagram. The glucose ones are quite diverse, and the phosphate ones are also quite diverse. And the English interpretation of this, I can't do it in Hebrew, but the English interpretation of this is that there are many ways to get better at phosphate and glucose, but only one or a very limited number of ways of getting better at sulfate. When we look in detail, and there's a fancier, since this is a bioinformatics school, there's a fancier way of looking at this, which is to take the Pearson distance between the entire pattern, and what you see is that you get one big peak, maybe a little bit of a shoulder. With sulfate, you get several peaks with phosphate and glucose. When you look at sulfate, what you find is that all, without exception, of the sulfate improved growers, the improved evolved strains, have an amplification of SOL1, which is the primary sulfate transporter. So they have solved their problem in a very simple way. There isn't a lot of sulfate. There's a limited number of sulfate transporters. We'll make twice as many, 3 times as many, 5 times as many sulfate transporters, and then we can grow. Whereas our sisters who didn't do that can't. Straightforward. What does this have to do with human cancer? Well, if you do exactly the same analysis for the genome-wide gene expression of human tumors, you find that different kinds of tumors are diverse in their human gene expression, but the breast tumors are all more similar to each other than they are to ovarian tumors or to whatever we have there, stomach, all these other different kinds of tumors. Again, in English, what that means is that the system that limits growth of the rogue epithelial cell that has to evolve to grow faster and faster, that the number of different ways of doing it is limited, and they're different for different types of tumors. Okay? So this is interesting. Again, we have an analogy. And in fact, we can take this further, and when we take it further, we can look at the breast tumors and the heterogeneity among them, and we can discern 4 different types, just like we saw 4 or 5 or so different ways of getting better at glucose. And the 4 different types are distinguishable on their pattern of gene expression. And better than that, okay, they are... when you look at what happened to the women who had these different types, you see that their clinical outcomes are different, substantially different. So these light blue, the dark blue ladies have a relatively low probability of having progression or death in 2 different data sets, and the magenta and the red have a much worse likelihood, and the light blue are somewhere in between. This now, after 10 years, there are several companies that use essentially this observation to guide therapy about whether or not you should do chemotherapy, because it turns out that these women don't benefit from chemotherapy. They get the side effects but no benefit. All the benefit goes to these. All right, so now we can do a truly genomic experiment. One of the students asked me about hypothesis testing. This is, up until now, for that student, wherever she may be, we have been doing discovery. Now we're going to test a hypothesis. Hypothesis is this. If there are several kinds of breast cancers, and they have different pathways of oncogenesis, then women who inherit a gene that predisposes to tumors, it's one gene, it should predispose to one of them. And we can test that hypothesis by looking at the patterns of gene expression of women who have that predisposition. And when you do that in a Dutch cohort, what you see is that all the dark red ones, all the BRCA1 carriers are the dark red ones. So the hypothesis has support. This is only an anecdote in this data set, but it turns out to be true that it looks like it's the dark blue ones that have BRCA2. And I'm going to skip the next one, which is about mice. If you look at genomic rearrangements in the evolved diploids, we keep getting the same kinds of results, and many of the reasons for these same kinds of results and the repeated things, partly the selective pressure, but partly also the mechanism of rearrangement. And in yeast, yeast has exactly 321 repeated transposon remnants. And we were able to show that many of these involve recombination within the transposon remnants to rearrange the genome. And these, we think, transposon remnants are not left around at random. The human has vastly more transposon remnants than yeast does. In fact, most of our genome is transposon remnants, it turns out. What we were able to see in yeast was that the recombination between transposon remnants, here's one piece, there's another piece, here you can see the deletion is right in between the transposon remnants, that these rearrangements you would expect that among the products of these rearrangements would be a circular DNA. And we can detect the circular DNA. I'm going to skip over the physiology of it. You expect that the recombination between the transposon remnant on the right and the one on the left would give us a circle, and that circle could reintegrate again, and also might, because it is a transposon, insert somewhere else in the genome. And so the expectation, which is we find, is that you will find occasionally these guys integrated somewhere else than whence they came. And of course, this is a hallmark. And here for the yeast biologist, there are the circles. And this experiment shows that the circle makes cells sensitive to dehistidine, which means that the gene from the circle actually functions. In humans, one of the hallmarks of cancer, especially for this particular oncogene, is copies all over the genome. And we think that they come from a similar mechanism. Okay, so let's see, how am I doing for time here? Good, I think. Okay. All right. So far, you can see where I'm going. Okay? So the problem with these experiments is that think about what we've done. What we've done is we have allowed evolution to happen in well-controlled circumstances. Then we've looked at one or two clones out of there, and we found things that we can interpret. That's very good. What we really want to know is more about the dynamics and the statistical kinds of things that happen in evolutionary time. And we know that these major things are nice to know, but there must be more going on. And so we turn to a different method. And in this method, we take advantage of some well-known yeast physiology. This is how yeast have sex. When they see one of the parents secretes a chemical that the other one recognizes, and then the mating begins when this recognition occurs. But in order for the haploid cell to recognize the mating signal, it has to express a number of genes. And the expression of these genes has a cost. And so if you just asexually propagate yeast, what happens is that one way in which they can succeed better than their sisters is to lose the ability to recognize, to turn on all these genes, and as a result, they can't mate anymore. They become sterile. And steriles have a growth advantage, as is shown here by careful measurements of somewhere between 0.5% and 2%. Now, the theorists who think about evolution in cultures made an observation. This is Desai and Fisher wrote this up very nicely. And when you have a small population, and the amount of variation in that population is also small, and you get a mutation that gives you a substantial amount of fitness advantage, like a sterile, then if this is an axis that has fitness as the x-axis, then you get moved over well outside the population in your fitness. You get this new mutation, and you have a huge head start, and the chances are that you will outgrow everybody in the vessel. On the other hand, if the population is bigger and or the diversity in the population is bigger to begin with, then it depends who you were, what your status was in the population when you got the boost of S and of fitness. And if you were in here, then the best that you can do is get near to the nose of the distribution. And however, if you were already here, then you get to outrun the population. Different situation. Okay? It has all kinds of implications. But before we get all run away with the implications, let's see if it's true. Okay? So, what we did was we hooked up this pathway to a fluorescent marker, and now we can tell when they become sterile not by a mating test, by taking the whole culture, putting it into a fluorescence activated cell sorter, and looking for dark cells. They would be steriles. And when we do that, we can make a calibration curve and show that we can reliably detect 95% of steriles in a non-sterile background or the other way around. And when we actually do the experiment, we get the following results. We get basically 4 kinds of trajectories. One is what we expected to begin with. A mutant arises and it fixes 100% of the population. Note this is a log scale. Then we get some in which these are other cases where we got that. We looked at 600 over 1,000 generations. Robotics were involved. If you want to know how, it's obvious, but I can tell you. Okay. So now what you find also are ones in which your original mutation started to go, and then another mutation, which was even better, happened, and what happens is this guy gets outcompeted. There are much more complicated scenarios that are possible, and then there's a kind of scenario that we didn't really expect, which we call frequency-dependent selection, where we get something which we don't quite understand. And if I have time, I can mention what that's likely to look at. All of these kinds of things happen in tumors. Okay? This is the whole data set. Now what we can do is we can test the idea of Desai and Fisher. So what you can look is at the slope of the trajectory and compare it to the fitness advantage, which is in green. And what you see is, in every case, the initial upslope is higher than just the fitness advantage. And so that is exactly what you would have expected from this. Okay? So, and here just shows you that as time goes on in generations, the average fitness goes down, not because the more fit cell is getting worse, but because the population is catching up. It's a race, and everybody's getting better. This is an artist's rendition of what this looks like, and this is what it looks like when we lose. Okay. Now comes the really cool part. Because of the advances in sequencing, and deep sequencing has become so cheap, we could ask the question, what if we looked in here? Since this one swept, there must have been another mutation that already fixed. And that mutation that already fixed was the one that gives us the extra head start that put it to the front of the distribution in the first place. And that was completely unselected by us, has nothing to do with sterility. It's just a mutation that happens normally that allows them to grow better. And we can now find that mutation by sequencing the whole population to high depth. And when we do that, sure enough, we find 2 mutations, ELO1 and GAS1. These are mutations that change the protein, so they're likely to be causal mutations. Those of you who follow the sequencing of tumors will recognize that we are now sitting in exactly the situation that the doctors who are trying to interpret these tumor sequences are in. They're trying to understand what's important, what's a passenger, what's a driver. But we have a huge advantage because we can find out. So we have ELO1, we have GAS1, and as usual with candidate genes, we can make a learned interpretation that in which fatty acid elongation was important enough, or this chromatin stuff was important enough. And of course, we found the mating mutation too, which we expected, and that was a nice frame shift, very satisfying. So which is it? So we take the mutations apart, take the strain apart, and it turns out it's GAS1 and not ELO1. So what is ELO1 doing there? Well, ELO1 is a passenger. What do I mean, passenger? I mean that if a mutation occurs before the mutation that is going to sweep, it doesn't have anything to do there, it's just a mutation, but then if it gets hooked, it hitchhikes on with the selected mutation, which in this case was GAS1, and it fixes. And we can also tell passengers from drivers in other ways by whether or not there's variation over time after fixation. And basically, we're learning the rules of how to tell the mutations that matter from the mutations that don't. Okay, I'm going to skip over the frequency of this thing. And I just want to end with the idea that actually, in studying these glucose cultures, we discovered something else, which is that just like the mechanisms that allow evolution to occur are conserved, it turns out that some of the results are conserved. And that if you look at the yeast, and I'm just going to go to the end here, if you look at what kinds of mutations give you the advantage in the chemostat, many of them are mutations in the same genes that are called oncogenes in various kinds in real tumors of humans, and furthermore, that like tumors, yeast which have dysregulated themselves, they also have this peculiar property of wasting glucose, which is called the Warburg effect. And this Warburg effect appears to be already present in yeast. So, I want to conclude by saying that there are striking similarities in the kinds of genetic events that you see in experimental evolution with yeast, that there are, let's see, for some reason, all right. Much of this behavior is because there's constant competition. The frequency of occurrence and the stability of mixtures of fitter variants, that kind of thing, which you see in tumors, you also see in yeast. I skipped over that. The number of paths to increased fitness is limited in the system sense. Okay, and you can see that analogy. And finally, that dysregulation of the metabolic control, okay, of cell growth results in glucose wasting in both yeast and humans. And finally, this is a summary of all the people who contributed to this in my group. And a particular thanks to George Weinstock at the Wash U Center, who has sequenced now many terabytes, because all of those 600 cultures we now have data for, and we're now sort of inching our way through interpreting it all. Exactly one of those cultures is fully interpreted. I told you about that one. The others, we know enough to know that many of them are similar, but there are many different genes involved, and we look forward to a catalog of all the genes that allow yeast to grow better by point mutation, as well as by these rearrangements and deletions and stuff like that, just like we hope to see that in cancers. And again, I want to thank the Dan David for the nice honor, and I'm happy to take any questions that you have. On time?
Yeah. Thank you very much. We have time for a few questions. There are two mics on the side.
Yes.
Yeah.
And the concept that change Yeah.
But can you say more about the link between the fact that you go mutation between asexual reproduction and sexual reproduction to the proliferation of cancer? It's not clear what's the end.
Both the evolutionary part and in both the tumors and in the yeast is asexual. Okay, I introduced sex because we used a trick that involves the readiness for sex to be the predictable visible mutation. It has nothing to do with the actual sex. In neither case is there any sex during the selection. Yes.
Any more questions? Let me ask a question.
Yes.
You talked about the mechanism of translocation in yeast. Can you say anything about the mechanism of aneuploidy? I mean, gain and loss of complete chromosomes?
In the experiments that we've done, a little bit to our surprise, we did not find the gain or loss of complete chromosomes, except we had some haploids that spontaneously became diploids. And I think that's a different phenomenon. It's a special yeast phenomenon, and we haven't studied it further. Others have published results that suggest that all the aneuploids grow less well than any of the euploids, and so maybe our naive expectation that we would find such things was simply wrong.
Okay, any more questions?
Yes.
You showed us the 4 groups of breast cancer.
Yes.
What about intragroup heterogeneity?
Okay. What about intragroup heterogeneity in the 4 subtypes? There is a lot of intragroup heterogeneity. We have not come to the end of the road because when we look at an individual woman and her metastases, they are still more similar than they are to others in the same group, although in some cases there are some ambiguities. And the answer is there may be more there than we have extracted in the way of information. We just don't know. We had took the big cuts, frankly, the cuts for which statistical support was easy to get. I think you'd have to do a fair amount more work to make those other differences truly credible.
Okay, let's thank Dr. Botstein again.
I'll leave this.